And I, on behalf of esteemed colleagues, want to welcome our global audience to our all-star conference here on cures or the quest for the cure of type 1 diabetes. We're honored to feature this group of esteemed colleagues, and particularly my dear friend and colleague going way back, Jay Skyler, who in this audience of people interested in diabetes needs no introduction. And by the way, we won't try to introduce all our esteemed colleagues. Those bios are easily found in the information about the conference. But Jay Skyler and I go way back. I remember, Jay, for example, back in 1993 when you visited me at FDA to talk about the diabetes prevention trial, DPT1. And we've been at it ever since, not just with insulin, but with other antigens, related and otherwise, and other approaches to type 1 diabetes. So Jay, thank you for serving as moderator of this panel.
I've done a number of these with the Kitalys Institute and the longevity series, Healthy Longevity, and I find each time interesting what are the things that we talk about, and they've varied the gamut. Today, it's been entitled "The Quest to Cure Type 1 Diabetes."
And Jay, before you do that, because we do want you to preview the seminar, let's first take care of a few housekeeping measures. And to do that, I'll bring forward my partner in many different ways, Thomas Sir, to give us just a few words before we get started.
Well, thanks,
Sure.
and a housekeeping reminder to enter any questions for the speakers in the Q&A function of our Zoom webinar platform. We also received questions at the time of registration. The panelists will try to get to them as time allows. As usual, a link of the recording will be circulated to all registrants and made publicly available within the next day or so. Just to highlight a couple distinctive features of our virtual webinars. One, we enable the chat function for audience interaction. So just for warm up, those of you who are willing, please say hi in the chat, your affiliation if desired, and from where you're logged in. Two, we will bring the formal segment of our webinar to a close at 12:30 p.m. Eastern Time, but we will keep the room open for 20 or 30 minutes for informal chat for those speakers and audience who can tarry. This is the virtual equivalent of the speakers descending from the dais for conversations with the audience after the in-person panel. Today's webinar is the quest to cure type 1 diabetes. The topic is particularly timely due to developments since our last webinar on this topic in July of 2020. I now turn the mic over to Kitalys Institute and Kinexum founder, Dr. Alexander Fleming. Zan?
Well, thanks, Thomas. No more is needed from me, but simply to hand over to Jay Skyler to preview our session.
Thanks, Zan and Thomas. The session is entitled "The Quest to Cure Type 1 Diabetes," and it is a quest which has been around for a long time. Many of the things that we talk about, I remember an ADA postgraduate course in 1973, in which one of the slides that was put up was headlines, and they included artificial pancreas, islet transplantation, all these things curing diabetes. We've talked about them for a long time. That's 53 years ago, and we've been continuing to pursue things. One of the things about science that I always find important is that you take 3 steps forward and then 2 steps backward. We look for progress, and we find there are new issues and new wrinkles that we have to consider. So it's not always a straight line. But the 4 areas that hold promise for people with type 1 diabetes is mechanical beta cell function, basically a fully automated insulin delivery system. I think we'll hear that we may be on the threshold of that. And that would make life easier for patients with diabetes, but they still have to deal with the devices, and it is a mechanical way of controlling glucose metabolism and mimicking beta cell function. The other three are trying to get biological cures for type 1 diabetes that we're moving toward. Preservation of beta cell mass and function is an important issue. A lot of that is by immune intervention to try to arrest the immune response. But what we've learned in recent years is the beta cell may be not just a victim of homicide, but a participant in potentially partially suicide, and that it's an interaction between the beta cell and the immune system that leads to the loss of beta cell function and mass. And we may need to have combination therapies to try to fully preserve beta cell mass and function. And we'll get to that during this program. Another issue that's just about to come into clinical trials, hasn't gotten there yet to any great degree, is regeneration of beta cells and trying to either get beta cells themselves to expand or cells to transdifferentiate into beta cells, or to get neogenesis from ductal cells that become beta cells, and that will be addressed as well. And finally, we get to replacement of beta cells. That's been around for nearly 50 years in one form or another, with pancreas transplants and subsequently cadaver islet transplants. More recently, trying to move toward stem cell transplants, but there are issues, challenges, and opportunities that exist in trying to bring those to fruition and to large-scale use. And we'll be addressing and discussing some of that. So we've got a relatively full plate as we move for our quest for a cure for type 1 diabetes. In looking at some of the questions that you and the audience turned in, everybody wants to know when, and is a cure possible, and when is it possible? And I would say that we're on that quest, but that quest, as I indicated, was there as early as 1973. We're now 2026. Can I give you a time frame? No. My hope is that I will live to eradicate type 1 diabetes and that we will get to this before my demise, but I can't otherwise predict things. And so with that, let's see. The next thing is that I will introduce our first panelist, who is Chantal Mathieu. She's from Katholieke University in Leuven in Belgium. She has been a pioneer in this space, having led 3 separate European collaborative initiatives, Name It, Enodia, which dealt specifically with immune intervention in type 1 diabetes and really trying to understand the nature of development of the disease, and more recently with a new operation. Why am I blanking on the name, Chantal? That you do with Identify, right, to identify people at risk of type 1 diabetes and see whether we can bring some of the potential interventions to them to try to prevent the disease. Chantal's been a leader of that. She's the most recent ex-president of the European Association for the Study of Diabetes and the most recent winner of the International Prize from the American Diabetes Association, where she spoke an address on that subject just a couple of weeks ago. So I'll turn it to Chantal and ask Chantal to please address immunotherapy and prevention of type 1 diabetes in particular. Chantal?
Thank you, Jay. And Zan said I should do this without slides. Are you still sure, Zan, that I cannot use slides? You're on mute.
You're on mute, Zan.
Chantal, if you have slides you want to show, then we're glad to do that.
Just share from your screen.
Yeah, I would like to share some slides because, as Jay said, trying to cure type 1, trying to prevent type 1, actually has been around for a long time. Actually, the title of the Toronto Times, the day after it became public that a child had been treated by Dr. Banting, said a cure for type 1 diabetes. And so it has been there for a long time, and indeed, insulin was then considered the cure. Of course, we know now 100 years later that when it comes to type 1, it was the slogan of the ADA, insulin is not a cure for type 1. It's there to treat people until we find a cure. And, JDRF and then Breakthrough also took these slogans over. But Zan and Jay, it is amazing what has happened in the last 20, 25 years on with our knowledge of what happens in type 1 diabetes. And it is not easy when you think back on how we had to do our research, because type 1 diabetes is a difficult disease. Everything happens in the pancreas, in the islets of Langerhans, well hidden, deep in the abdomen, unreachable. The dermatologists who study vitiligo can take skin biopsies. Rheumatologists can take biopsies of the joints or puncture the joints. Even doctors studying MS can, do a puncture. For us, we cannot access the organ. And so that has been a challenge. We always have to use biomarkers for the disease activity. We also need to use measures for the functional beta cell mass. We have stimulated C-peptide. We all agree it's a good measure of the functionality of the beta cell, but still we do not have the histology until we can take out the pancreas of somebody. But that, of course, is not compatible with a life without diabetes and further on. And so we had to rely on animal models, the NOD mouse, and many people criticize the NOD mouse. But still, it helped us to come to therapies. And we're living in very exciting times now, because we do have our first disease-modifying therapy that has been approved by FDA, by EMA, by other instances all over the world now, namely teplizumab, an antibody directed against specific T lymphocytes that we know play a role in the destruction of the beta cell. The drug called teplizumab, indeed, approved in stage 2 type 1 diabetes, so the stage where we know that the destruction of the beta cell is accelerating, leading towards the stage in the disease where insulin is needed. Has now also been approved by the FDA very recently, a couple of days ago, for use in people with stage 3 T1D, so the stage where insulin is needed, because we also have enough evidence now to agree that if you treat people with newly diagnosed clinical stage 3 T1D and you give the teplizumab, you can still delay the further destruction of the beta cell. And we know that by measuring the functional beta cell mass by stimulated C-peptide. So we have our first disease-modifying therapy. And from other diseases like rheumatoid arthritis, we know that once you have a foot in the door, there's a whole world that will open. But so I just want to share some evidence, and especially also some overview. And this is a slide where you have an overview of investigational agents that have been studied or are being studied in people with clinical stage 3 type 1 diabetes. We have indeed, if you look at our, as I call, the lethal circle of destruction of the beta cell, with cells of the immune system like T lymphocytes playing a crucial role, cytotoxic T lymphocytes destroying the beta cells. But what we know is that also inflammatory cytokines play a role. We also know that, for instance, other cells like neutrophils and K cells are playing a role. But still, when you look at targets in this circle of destruction of the beta cell. We have quite a few. I already noted the first approved agent, teplizumab, but we also have evidence, for instance, from the MELD-ATG trial, and I'm sure, Zan, you will want to discuss this because it was an innovative trial design designed to make the field go more rapidly, testing different doses. We know that anti-thymocyte globulin, old drug used in transplantation, for instance, if we use it at very low doses, we can indeed also interfere with this circle and of destruction and make the beta cell go down in mass and in function more slowly than if you do not treat people at all or if you give them placebo, but also other agents, and interestingly also agents that do not primarily attack the immune system, but also make the beta cell stronger, like verapamil or liraglutide, for instance, a GLP-based therapy, can interfere with the circle, and we have evidence there. And I just, want to salute the approval of teplizumab here. This is my last slide. I want to show Thomas and Zan, just to show to the audience that there's also a lot of agents being tested now in the early stages of T1D, because indeed we know now, and I want to go back to another slide, because this is now how we consider type 1. When most of us were in medical school, we would say, type 1 diabetes is a disease that starts quite suddenly and insulin is required for survival. We have now completely reclassified this. We say that this is stage 3 T1D, namely when people need insulin. But we say that the disease itself, this autoimmune destruction of the beta cell, is actually starting many months, many years before this clinical stage in many people. And we call that stage 1. And so stage 1 is the autoimmune attack is happening. And we know that because there are markers of the autoimmune attack in the blood of people, namely autoantibodies against insulin, GADs, zinc transporter 8, etc. And the intermediate stage is stage 2, where you have the antibodies and already more than enough beta cells destroyed to have dysglycemia. So this is the new phase of type 1 diabetes. And I showed you the interventions in stage 3 when insulin is needed. 20 to 30% of your functional beta cell mass is still there to be rescued, but it allows us to make proof of concept trials. But what we really want is to intervene early on when there's still enough beta cells left to maintain normoglycemia and not need insulin. And so this, for instance, is in stage 2, and that's also where we have the approval of teplizumab. But also other agents are being tested as we speak in clinical trials. But so, Zan, a lot is happening. Things are moving very, very fast, but not fast enough. And I guess we will discuss later on novel trial designs to speed this all up.
Well, merci bien, Chantal. That was beautiful. The one, with the exception of GAD, there are really no examples of tolerizing approaches, which actually Jay initiated with the DPT1, using insulin itself as an antigen that might tolerize against diabetic autoimmunity. And so that has been a very challenging area. Jay and I were involved with another effort that involved a plasma-delivered proinsulin approach. And yet we just fall short of any success with approaches that might have an even better safety profile than these monoclonal antibodies that do take out part of the immune system, at least transiently.
Yeah, if I may reply, Zan, I think we are delusional if we think that intervening with an antigen in a late stage, when this autoimmune attack is heated up. It's like coming in a kidney transplant during a rejection. I think we're delusional to think that if we come in at that stage with an antigen alone, like insulin, or even an antigen like GAD in an adjuvant like alum, that by just doing that, we will be able to arrest the beta cell destruction. So I am a believer that antigen-specific interventions may help in adjunct with immune modulation, but there's an opportunity very early on in life, before the circle of destruction is active, in genetically at-risk individuals. And that's, for instance, what Annette Ziegler has tested and is testing in people genetically at risk of type 1 diabetes. So before any autoantibodies are present. And then perhaps we have a window of tolerance restoration just with antigen. But that remains to be proven. The evidence is not fantastic at the moment.
Right. Well, a lesson learned. I think you make a compelling case that that at least is something that we now understand, and we just have to seek alternative approaches. Well, again, it was a wonderful, all too quick review, Chantal, but we'll come back to you, and let's now move on to Susan Bonner-Weir. Susan, you actually, along with your dear late husband, have been preeminent in the field of islet biology and seeking to understand how we could restore normal beta cells, or at least insulin secreting cells that are preserved in people with early type 1 diabetes. So really looking forward to your overview of that field and what we might expect to come out of it in terms of actually regenerating beta cells.
Thank you, Zen. Actually, I'm very happy speaking on this topic, but I think the first thing I need to say is you're not in type 1, no matter how much you can regenerate islets. If you don't have the immune system tamed so it's not autoimmune, it's not going to do much good. So we really need sort of teamwork with the immunologists before we're going to get to anything that anybody could really call as a cure. But I would like, if I may, just to have a couple of slides, just because, and if I go to it, wait, I'm sorry. There, can you see it?
Not yet.
No, I guess, I'm sorry. Well, basically.
No, take your time, keep going. We're glad to wait.
Go to share. There it is.
There we go.
Now you should see it. And now I can put it on the slide.
We got it.
Yeah. So really it's sort of a dream to get the beta cell regeneration. I am a firm believer that the pancreas normally is having growth throughout life. And there are various determinants of what the amount of beta cells you have left or at any one time, and it comes from how many are replicating, how many are turning over and have a slow sort of loss of a beta cell. They all embryonically are coming from the same embryonic endoderm that becomes pancreas and then ends up differentiating into the different cell types of the pancreas. As sort of displayed here, they're really there's sort of 4 ways or 4 sort of cells that you could imagine that you could get expansion of or differentiation of to replenish the beta cells that are being destroyed in type 1. There's replication of beta cells of the pre-existing cells, and even at onset, people have using the NPOD pancreas samples have shown that at onset you really do have maybe up to 30 to 50% of the pre-existing beta cells are still surviving. And possibly what's happening with the honeymoon is that the honeymoon that one sees after insulin treatment is started is that the beta cells that are left are now sort of have lower glucose, hyperglycemia, so they now can function better. So it's a balancing of physical cells as well as they're being able to function properly. So you can have a normal sort of a low level of replication of beta cells. What I have worked many years on is that you actually, I think there's the thing called neogenesis, which is new formation, newly differentiating beta cells. In the context that we've done, it's really from a duct cell. Some people would call that transdifferentiation, but I actually think I've always called it more that the duct cells dedifferentiated and went back to their sort of set progenitor stage and then could move forward to forming any of the other pancreatic cell types. There have been some interesting studies with rodents that have really shown that you can get sometimes with coaxing with sort of by gene editing, by giving adenoviruses, and sometimes just by making the situation very severe, for severely loss of beta cells, you get either other islet cells or acinar cells to become insulin producing cells. It's at that sort of was very exciting when the rodent stuff came out, but it seems to be a very slow process, and I'm not sure how much has progressed with that. The other thing that one always thinks about is whether there are actually true stem cells within the pancreas, and I have not seen evidence that anyone's reported that they're actually true stem cells. I think they're more sort of a progenitor cell, and there may be particular cells that are remain in the adult pancreas that are a progenitor, but I don't think they're a true stem cell in the after birth. I think one needs to appreciate that in the adult human pancreas, there does seem to be a very low level of expansion or turnover, as it were. There's replication. When you do it from an autopsy, you don't see any. In fact, when I got into the field, I think people basically thought that in the adult, the beta cells do not replicate. But I think that's been clearly shown, even in humans, that's not the case. In surgical specimens, or when you've transplanted human islets into a mouse, like under a kidney capsule, you can see sort of 0.5% of the cells are in cell cycle and actually can be labeled that they are replicating. That would take a long time to get where you need to be if someone already had type 1 diabetes. But the other thing really is that has had a lot of work is the neogenesis. And some people, hopefully they're now realizing that what we see in the human pancreas is not exactly what one sees in a mouse pancreas. We learned a lot from working with the rodents, just as Chantal said, for the immunology, but the human pancreas has a number of differences, and one of which I think is the frequency of what we call neogenesis, such that in human pancreases, first of all, you tend to see an increased number of insulin positive cells within the ducts, or with also co-expressing some of the duct markers, such as the cytokeratins that are characteristic of the ducts. But the interesting thing is that over the last maybe 10 years, there have been more and more studies that have actually started looking for it in human pancreas, and have actually seen and quantitated that you have increased insulin positive cells in the ducts, or that when you've had pregnancy, insulin resistance, the early stages, the progression going from impaired glucose tolerance to type 2 diabetes, or more recently, there's even been a very nice study that was showing that you had more when you had sort of local injury. So I think that we know that it can happen at a very low rate. In fact, I think the two things that I think really have convinced me that it is possible to get neogenesis in the human is the fact that that after birth in the human, you have an incredible growth of the pancreas. On the left is just the pancreatic weight, which changes from birth to young adults, the age 25. It's sort of almost 50-fold increase in the volume of the pancreas. And the mass of the beta cells at that point really increase sort of 8-fold from birth, even though at the time it's really interesting that the proportion of beta cells does not change after about the first year or so. The proportion of beta cells within the pancreas, the relative volume, is basically constant after sort of infancy. It's low, but it's constant. And when you have this much growth, there has to be sort of expansion, not just of cell size or replication, because most studies have shown that there's not a lot of replication after the 1 year of age. It is still also, it's a low level. But what I think what ends up happening is that you're getting the growth of the pancreas in length and width, and you're getting new lobes of pancreas forming, and with that, you're getting the neogenesis, that you're getting new islets forming as you're getting whole new lobes formed. So I think that that's what my mind, that there is a period of time, even as you see, even into adolescence, you're getting increased growth of the beta cell mass. So I think that we have that opportunity that there's the potential for stimulating beta cell neogenesis at that point. The other sort of thing that's really convinced me has been with the medalist studies. George King started a study of the people who had gotten the 50-year of insulin-dependent diabetes medal, and has really done extensive studies on it. They are, for the most part, typical type 1. They have the right HLA. They have only a few have very low levels of C-peptides. Certainly they are, and many of them do have, are antibody positive. But over the years, we've had 86 of the pancreases that were donated after death, and the average of the duration of type 1 diabetes was 67 years. And yet, every single one of the patients, of the donors, had insulin positive cells that have survived with at least over 50, and as I said, a mean of 67 years of type 1 diabetes. So everyone has, not very many, but they do have insulin positive cells. But what I've really been struck by is that a fifth of them, 22%, have lobes that have islets, as over here on the, I guess it's the far right, that have significant number of beta cells, and there be the whole lobe maybe, and there may be 2 or 3 lobes. It's still a very small fraction of the pancreas. But the medalists are all over the age of, most of them are, well, most of the pancreases that we've seen have more or less all been over the age of 60. So to me, this seems like it cannot really be that they are just residual from all this, from their whole lifetime and the long duration of type 1, but rather that there is a continual growth and a sort of reformation or formation of beta cells and continued autoimmune insult that wipes them out. In fact, with some of the antibody studies and the C-peptide studies, you would expect the going from a positive to negative, but in the red boxes as shown here, there is a percent, around 10 to 15% of the people who were studied living when they came back 3 to 5 years for a second testing, actually showed that they went from negative antibody to positive antibody, or negative C-peptide to positive C-peptide. So I think that also sort of supports the idea that you're having, even in medalists, even in older people above the age of 50, you're going to have some potential of turnover. So then you get to, okay, so if you have turnover, what's happening? What have people been able to show? Well, a lot of the things that worked in rodents didn't really carry over and work in humans. This has been particularly disappointing for the things that people had found for stimulating beta cell replication. But what has been, again, it's probably, again, the 10-year time point when people started focusing more and had the better availability of being able to get tissue for study from humans, whether it's isolated islets through various organizations like the IDDP, or just slides from organ quality donors such as NPOD. But what people have been able to show, and particularly I think Andy Stewart was one of the ones who really showed that you could, with harmine, which was a natural product, they could show that in vitro that you could get, or also transplanted into rodents, human islets, you would stimulate replication up to maybe 10-fold increase. Now, harmine has a lot of problems that affects the cognitive, the brain, and I think hallucinations and whatever. So it's not exactly your ideal treatment, but they found that the mechanism of harmine seems to inhibit the DYRK1AK, and there was a clinical trial using different doses of harmine to see how safe it was. They did find that the lowest doses, you didn't have the side effects. I don't know that they've been able to show that actually had any efficacy on replication in vivo. but there are a number of compounds that are being tested. And I think some of them are also starting to be tested in clinical trials to see, again, first they have to do the safety, but there is potential there. And I think that's important because even if we get new formation of islets, you probably would want to have stimulation of replication in addition so that you can amplify what you have gotten newly formed. So again, not only we need the immunologists, we need the focus on the replication, but the third thing is what I've been particularly interested in is, are there things that can actually stimulate new beta cell formation, the neogenesis aspect? And I think the Miami group has done quite a bit looking at and finding what they think is a progenitor that's sensitive to BMP, bone morphogenic protein 7, BMP, or I think they've recently used agonists for that receptor and have seemed to stimulate new islet formation. At Joslin, Pong Yi has a paper that should be out very shortly that did a screen for knocking out, it's a loss of function, did a screen and found, I think, eight genes that if you knocked them out or down, that you ended up getting insulin positive expression. And one of these they followed up on, which is an aldehyde dehydrogenase 3b2, and actually used that in human purified primary human ducks, exposed those to lentivirus that had both the mechanism so they could delete this out of this gene. So it had the Cas9 and guide RNA for this gene. So using sort of a CRISPR technology could get this mutated duck cells. And when they transplanted those cells into diabetic mice, within 3 weeks, you were getting insulin responsiveness, human insulin responsiveness. And by 6 weeks, the blood sugars were almost normal. And yet they only had about 15% of the cells in the graph that actually had been mutated and expressed insulin. Now, 40% had been mutated, but only 15% were able to express insulin and the other sort of identity genes of beta cells. So it's inefficient, but it's very exciting in my mind that you actually are getting studies like this finally. How long it will take to get from here to where you can actually do the clinical trial, that's I think the BMP7 agonists are probably a lot closer, but I think it's really it's exciting at this point that we finally do have a few molecules, both for replication and for neogenesis, that seem to have some potential. So
Great.
Again, it's a matter of needing to have different things come together. The testing of the neogenic factors, testing of replication, but mainly keeping the autoimmunity tamped down so it does not destroy them.
It's a recurring message. We need combinations. And I like the title of your slide, The Dream of Beta Cell Regeneration Still to Come. We're going to move on. to the next presentation, next discussion. And Peter Senior, who is a professor and the director of the Alberta Diabetes Institute in Edmonton, in Canada, is going to talk to us about transplantation and manufacturing of new beta cells and how we go about thinking about that. Peter?
Thanks, Jay, and others, and thank you to the audience for attending. My challenge will be not showing slides, but containing how much I say. But I'll try to kind of focus on the things which I think are new and relevant. So my expertise has been, developed over the last 20 plus years in deceased donor islet transplantation. And a lot of my colleagues, when I began this, said, well, that will never be the answer. There will never be enough beta cells to treat all the people with diabetes, and there's all these drugs, and isn't it terrible? And that is in some ways true. The two major barriers are we're never going to have enough donor islets to transplant all the people with type 1 diabetes, and currently we need to use systemic immunosuppression. But for people where that risk benefit makes sense, islet transplantation is transformative. And it's been a huge privilege for me to work with people over the years who can't be on their own, who can't sleep safely in their beds at night. And we can give them a treatment that allows them to live life free, with flexibility, able to do the things that you and I take for granted. The kind of dream in some ways of being able to make islet cells or beta cells that you could overcome this supply issue has felt like science fiction for many years, and yet it is now science fact. So if you think about all of the science which has gone into understanding how embryos develop, how cells which have got all this potential to become any cell in the whole body, how they develop and grow and choose to become a pancreatic cell and then become a beta cell, that science has been worked out very, very carefully. And so now we have a somewhat complicated but equally well described recipe to make that happen. And we have got a range of sources of stem cells that can be used to generate beta cells. We have embryonic stem cells, which are the ones which maybe people think about most often. But we can take adult cells, and maybe as Susan alluded to, we can sort of unprogram them, take them back from the mature state to childhood, to infancy, to become these pluripotential stem cells. And on the one hand, we could do that with an off-the-shelf line that we use to make cells for everybody. But the other approach is to kind of bespoke, custom design a cell line from an individual. And so this situation where you could take somebody with diabetes, take some cells, maybe fat cells or skin cells, differentiate them, then train them to become beta cells or islet cells. That is now possible. It's expensive. And to do that for each individual person with diabetes, again, may be sort of prohibitively in terms of generalizability and rollout. But nonetheless, it's very, very attractive as a prospect. Let me just draw a bit of a contrast between what's been happening with deceased donor islet transplants and what's happening with these manufactured or stem cell derived islet cells, which are now in phase 3 clinical trials. When we have a donor pancreas, that donor has died almost by definition. And so the process that the pancreas has been through, going through that death, then the harvesting of the organ, the processing, it's a pretty traumatic experience for the islet cells. And we don't have a process that's 100% efficient. And so for each donor, we can maybe extract around about half of the cells that make hormones. When we implant those into the liver of recipients, we probably lose another half. And so you're then getting quite a marginal mass of islet cells and beta cells in that donor, in that recipient. And so generally speaking, we need 2 or more doses of islet cells to get people over the line so they don't need to take insulin injections. What has happened with some of the stem cell derived products that have been transplanted into the liver with anti-rejection drugs, very much as we've done with deceased donor islets over the last 20 plus years, those cells engraft. It takes them a few months to actually fully mature, that they actually start making insulin in response to glucose. But those cells will enable people to become insulin independent with 1 dose. So rather than needing 2 doses or more, 1 dose of these stem cell-derived products can achieve insulin independence. And it's intriguing to think that these are brand new baby cells that have got a lifetime ahead of them. The deceased donor islets, which I give to patients, are secondhand. They've lived a life already. And so that may be an important thing to consider. But still, we have got this issue of anti-rejection drugs being required to stop the immune cell attacking these islets. And so many people, much smarter than me, have thought, but what if we could make these bespoke stem cell derived islets that matched the recipient? Take my cells, make them into islets, give them back to me. Wouldn't that be the answer? Because then there would be no rejection of a foreign tissue, a foreign cell type. And so this work has been happening and has been published in several patients from China. The 2 initial reports describing this were really very, very exciting. But again, because this is experimental and we're trying to maximize safety as well as effectiveness, those procedures were performed in people who had already had other organ transplants, either a liver transplant or a kidney transplant. And these patients were already taking anti-rejection drugs to look after those other organs that they had. But those, what we call autologous stem cell derived islets, enable those patients to discontinue insulin. And again, taking what in my head had been science fiction and making it science fact. But the question that was still lingering in my mind and the minds of many, many others was, but do you need the anti-rejection drugs? What happens if you did that for somebody with type 1 diabetes without immunosuppression? And what I'm led to believe is that when that was attempted, again, taking cells from somebody with type 1 diabetes, making them into islets, giving them back, without immunosuppression, the autoimmune process rejected those islets. So those people's immune system, yes, it doesn't like foreign tissue, but you know what? It really, really doesn't like beta cells. Doesn't care whose they are. Yours, mine, somebody else's, they rejected that. And so that is a big, big challenge. But I think there's hope. If we think about immunosuppression or immune modulating drugs as a massive spectrum, there's a kind of full body, full dose things we use in organ transplantation. And then there's the drugs that you see advertised on TV for psoriasis, for lupus, for rheumatoid, that people are taking and they're living their lives. And again, it may be that some of those lighter immunosuppression approaches may be what we need for these stem cell derived products. So I think I'm going to pause at this point. Again, there's lots more to say. We want to have plenty of discussion, but there is real hope. And science is moving very rapidly, more rapidly than I expected, which is unlike much of diabetes, it seems. We've waited 100 years, and are we that much further on? But I think there's some real progress that I hope is diabetes turn for that kind of quantum leap that we've seen in other disease states.
Well, thank you, Peter. That was superb. And we'll come back to some of these points in our general discussion. I now want to go to Greg Forlenza, who is from another hotbed of type 1 diabetes research and development, and recently a camp doctor for kids with type 1 diabetes, returned from that. So Greg is very active on multiple fronts, not just diabetes technology, but for example, leading the trial of verapamil for people with new onset type 1 diabetes. So Greg, thank you so much. And we look forward to your presentation on the diabetes technology, which has made such great progress.
Yeah, thank you, Zane, for that introduction. Thank you, Jay, and to the committee for inviting me. And thank you to all the previous speakers. My voice is a little scratchy today. I was at our Camp Colorado last week, and in caring for the kids for the week, I got sick, and I have to reflect on the fact that the 14-year-olds got better in about a day. And now that I'm in my mid-40s, it's taking me about a week. So I'm sure that'll get better over time. And so thank you guys for your interest. I also was a little hesitant about not creating slides, so I went ahead and created a few slides just to talk about this. And so as I was putting this together, I kind of felt like I was sort of the odd duck out in this, this group. Everyone else who's spoken so far has talked about what I think any normal person would think are the ways that we're going to cure diabetes, that we're going to modulate the immune system, we're going to get in before the immune attack starts and suppress the immune system attack of the cells that make islets, we're going to transplant the cells that make islets, we're going to regrow the cells that make islets. And here I am talking to you about computers. And so why does this make sense? And I think the reason that it makes sense comes from the argument about the sewing machine. I was hesitant about putting this in here because I think this is going to be a little out there, but I did want to share this. And so my maternal grandmother was a seamstress. Growing up, we had this old iron Singer sewing machine in her house that she taught us all to use. And anyone that has ever studied a sewing machine has realized that the thing is absolutely insane. During the Industrial Revolution, as we were trying to solve the problem of automating various tasks that humans were performing, one of the hardest things for humans to automate was the process of sewing. And the reason that this process was so hard to automate is that the people trying to invent the sewing machine were trying to invent a device that replicated the way that humans sewed naturally. Anyone that's ever sewn a button back on their shirt knows that there's a process of passing the needle all the way through, pinching, pushing, grabbing, turning, pinching, pushing, grabbing, turning of a needle. And that was essentially impossible to do with 18th century technology. If you think about programming it, it would be hard to get a computer to do it now. And so by trying to replicate the way that humans did the process naturally, we were unsuccessful in building a machine that could sew. The revolution came when the inventors completely abandoned the rational approach and decided to invert the needle, put the thread in the eye of the tip of the needle rather than the heel of the needle, add a second strand running perpendicular to the main strand, and turn part of the device upside down. No part of building a sewing machine sounds sane. However, every article of clothing everyone listening to this talk has ever worn in their entire life has been produced by this method. It is backwards, upside down, crazy, insane, and it is the best approach to sewing. And obviously you can see where I'm going with this. This is the type 1 diabetes artificial pancreas roadmap laid out by Aaron Kowalski in 2009. The idea behind this was to find a technological solution to insulin delivery that we could build using tools that existed at the time. He then updated this in 2015 to a slightly different schema, but this has been the guiding light behind technology development over the past 15 to 20 years. And this process is not a cure. We all agree that it's definitely not a cure. But what we've been able to do with this process is make each part of the sewing machine, get it to work, add in the next part, and get that to work in the same way that we build or innovate anything. everything the previous speakers talked about today is way more complicated, way more advanced, and way more scientifically difficult than any part of this process is. And so what have we achieved by doing it with the technology approach? We've achieved this, where in the last 10 years, we've gone from a roadmap of things we weren't sure we could build to multiple different CGM systems on the market in the United States, to multiple different automated insulin delivery systems on the market, multiple different interactions of these devices, and new devices coming so quickly that I have to update this slide every time I give the talk. And I always tell people, I can't even keep this straight, and it's the only thing I do. That's how quickly we're moving with this. What result have we achieved with this? So this is cross-sectional data from our pediatric clinic. I'm a peds endo, so to me, diabetes means taking care of children. I acknowledge that there's former children, currently called grown-ups, with diabetes, but I focus on the kids. And what we've achieved in the past 10 years of this development is in our clinic right now, 80% of our kids, this is all the kids in Colorado, public insurance, private insurance, diverse racial ethnic backgrounds, diverse socioeconomic backgrounds, 80% of our patients are currently using automated insulin delivery systems. Those kids have a hemoglobin A1C of 7.8% on average, which is down from about 9.5% just 10 years ago. And this is just Gen 1 of the technology. If you take a step back from, the focus of what we're talking about today, what is the thing that humans are exponentially gaining knowledge about right now? Well, of course, it's neural networks, it's AI, it's the way that we use computers to build and run large data-driven models. And we were talking about ways that we can use data-driven models to analyze data on immunology and epidemiology. But this is the way that we can use a data-driven model that uses neural network processing to essentially make diabetes decisions. And the reason this is so powerful is that it leverages very large data sets to make very accurate decisions in a quick iterative fashion. And as most people are aware, we've gone over the last 5 years from this being something that sounds like it comes out of an Asimov novel to something that is recording this talk while we're speaking today. And this is where we're seeing exponential growth in human knowledge right now. The question a lot of people would have is this crazy or does this work? And the answer is, it works. Our group has done studies on this in adults, adolescents, children, and young children, where we've demonstrated that AI-based fully closed-loop controllers can basically maintain time and range values for people with well-controlled diabetes, similar to them doing their best, without them needing to interact with the systems. We can take people who are not currently doing well and see significant gains with less engagement. And so where are we going with this technology? This is one of the studies that we're running right now. The first company that commercialized automated insulin delivery, Medtronic, or now MiniMed, is running their FDA approval trial for their fully closed-loop controller, a system that doesn't require any bolusing, a system that essentially has no tunable parameters other than a target. We're running it right now. Part of the reason that I wanted to introduce being at camp last week, other than I love camp and that's why I got into the field, is that I had people wearing this system at camp last week where their time and range was over 70% when they were at 9,500 feet, exercising 8 hours a day, eating terrible camp food with an irregular schedule and not bolusing, and having over 70% time in range with a system that requires no tuning. And I think this is really exciting. And what I say to my friends who are working on the solutions that sound rational is, I hope you beat us, but we're going to give you a really high bar to clear. And I think that is a good goal for us to have as a field, that our biological solutions need to be solutions that can beat something like this, because we have this right now. I have kids at home right now wearing this system, and it's something that's going to come to market within the next 12 months, and not just from one manufacturer. We're running trials right now in the Omnipod 6 automated insulin delivery system, which we're testing in fully closed loop mode, and we're designing the trials right now for the Tandem Control IQ successor, the AdaNet system, in fully closed loop trials. And these are things where people would say, you know, when is this coming? And I respect all my colleagues who say, I don't want to give you a timeline. But for technology, we've done this before. We've done this a lot over the last decade. We will see fully closed loop systems on the market in the United States in 2027 and 2028. It's not that far away. And the idea is it's not that hard. And it's the thing that humans are gaining knowledge on exponentially. And so there's also a whole lot of, you know, help using these systems to help islet engraftment, or using these systems to help breast islets as you're doing other therapies. I don't think these things are actually competitive. I think they're synergistic, but for debate, it's fun to compete, right? And so, but that's the idea, is that these technologies are moving very quickly, and that while they are certainly not the intuitive cure that we think they should be, they may just be the sewing machine that is a better solution than we thought we'd come up with naturally. Thank you for letting me include the thing about my grandma on the sewing machine. That was fun.
I love that metaphor, Greg. And wow, very compelling message. You've clearly shown not just the promise, but the actuality of technology for making the lives of people with type 1 diabetes much better. One sort of dark lining is that it makes it that much harder to develop disease-modifying therapies if everybody is using these closed-loop systems. That is, to show clinical benefits in people who are using any kind of CGM or insulin system or insulin pump system. So we might come back to that as part of our challenge, but the glass is much more full than empty when it comes to diabetes technology, and you've clearly shown that. I would like to turn back to Jay and Chantal before we go into our general panel discussion about how we can speed the development of products that will make the lives of people with not only established type 1 diabetes, but those at risk much better. And this comes down to what can we do to improve the regulatory evaluation and pathways for getting products on the market. As some of you may know, Jay, Chantal, and I recently published an article that advocates for C-peptide as the regulatory endpoint to lead to full approval of products for new onset type 1 diabetes. Now, this doesn't work for other applications, like for cell therapies, which need to show clinical benefits to be approved, nor does it work for products that are aimed at preventing clinical diabetes, type 1 diabetes. But Jay and Chantal, let's come in and talk about how we can improve the climate for developing these disease-modifying therapies, both opportunities and challenges.
Well, Zan, I would modify your statement a little bit, because I think even in prevention or in replacement of beta cells, we measure beta cell function by C-peptide. And so C-peptide is going to be an important component of any of the strategies that we're talking about, except for the mechanical approach that Greg approached. I think whether it be regeneration or neogenesis of beta cells, we'll use C-peptide as an indicator. We're going to want to see preservation of C-peptide in the prevention phase, and in replacement of beta cells, we're going to want to see C-peptide there. And it's been demonstrated in islet transplant that even if you don't have insulin independence, if you have maintained C-peptide, you eliminate the recurrent severe hypoglycemia that Peter was mentioning.
Right.
Yeah,
I think
yeah. Can I just
ahead.
build on the remark you just made to Greg, saying that the novel technologies, especially once we move from hybrid closed loop to fully closed loop, will make demonstrating added value of disease modifying therapies more difficult. And that is true when you define success of an intervention as when you do the intervention in people in stage 3 T1D, so needing insulin by the hybrid or fully closed loop, if you define success by showing superiority on hemoglobin A1C or dramatically reduced insulin doses for a similar hemoglobin A1C, it will become challenging, especially when we need to show superiority in hemoglobin A1C because our technologies are so good. But again, the point we try to make is this is a disease of the beta cell. It's the immune system destroying the beta cell. So if you can show that your intervention treatment is interfering with the destruction of the beta cell, namely that function of this beta cell is preserved. And again, I think we made the point that stimulated C-peptide is really the measure of the functional beta cell mass, then we are out of trouble. And so that is the challenge, really, to convince the regulators that in a disease where we have now our fantastic technologies showing superiority in hemoglobin A1C once you go into people in stage 3 T1D is not what we should be demanding of the disease modifying therapies, but rather show me that you interfere with the disease progression. And
right.
So
what I would like to see us do is not only rely on C-peptide, but with disease-modifying therapies or combinations that end up allowing the patient not to need insulin at all.
That's another thing. I mean, yeah.
In that circumstance, the disease-modifying therapy doesn't need the mechanical insulin delivery and doesn't need to beat that.
Absolutely. And that's what we all dream about. That is that we can have an intervention where we have what Susan was suggesting, namely proliferation or regeneration or whatever of the beta cell, or a product that makes beta cells stronger and laugh in the face of the immune attack, and then one or other immune modulatory agent. The problem is that our regulators demand that we show effect of one, effect of another, and effect of another. And so that will take us years of single therapies showing something before we can combine. So we have work to do with the regulators, convincing them, first of all, that indeed stimulated C-peptide is the measure of functional beta cell mass and thus can be used as endpoint in stage 3 trials. Second, that we can propose from the beginning combination interventions, and then also novel trial designs like what we did in the MELD-ATG trial, an adaptive trial design testing different doses. We went using ATG from 0.5, 1.5, 0.1, 0.5, 1.5, and 2.5 milligram of ATG per kilo, all in one trial, with a dose determining committee dropping arms. That way, you can test in 1 year 4 doses of one disease modifying therapy, because until now we do one dose, test it, oh, it didn't work, do another dose, test it, oh, it didn't work. So adaptive trial design, platform trials where we test different interventions compared to each other. So a lot of work. The good news then is that, for instance, in Europe, EMA is listening. again, identify the project looking at early stage T1D. We just introduced a new master protocol now proposing platform trials in people with stage 1 and stage 2 T1D, where the endpoint may include C-peptide, but also have other metabolic parameters as endpoints. So we have work to do, but I do believe we are progressing.
Well, indeed. And Chantal, I hope my former colleagues at FDA will follow their colleagues at EMA in being open to these various approaches. And of course, adaptivity has been long, and platform trials, these have long been used in the oncology community. There's nothing new about that. We should be using them. And go beyond that. I believe there is an even more streamlined approach that can be proposed that will.
I think Peter wants to say a word about non-inferiority trials, and then Greg has a comment. Peter?
Right, and go ahead, Peter and Greg, and we'll broaden our discussion here.
I mean, I think, Greg, your points are spot on in terms of, glycemia being something that can be achieved a lot of different ways. But I did reflect at the kind of technology conference last year or the year before. It's kind of like, I'm so excited about all these toys, but is that really what my patients want? They want not to have diabetes. And it may well be that if we have this glycemic focus, that we'll have to think about non-inferiority trials. We may even need to be thinking about not just non-inferiority for A1C, but also for hypoglycemia. And it may be those kind of, patient-reported outcome measures that may be the really important thing. And it seems to me that often providers, payers, regulators underestimate how burdensome living with type 1 diabetes is. And again, the ability to sort of restore that independence, that freedom, may be something that's very, very valuable for people with diabetes. And again, I would like to help develop tools that can sort of measure that, because that could say, yeah, these give you the same A1C, the same time and range, but this one, you can shower naked every day, for example.
Excellent, Peter. Greg, go ahead.
Yeah, so I agree with everything Peter was saying there. I love the technology work. I've been a technology person my whole life, and I like to advocate for that. But the first thing is, in terms of the technology making the biological approaches unnecessary, there are natural limits to what we can do sub-Q. All of us acknowledge that. I mean, no one that I work with in the tech space who develops this technology thinks that we'll be able to get to biological normality. It's just, can we get to a really awesome floor? Where the worst case scenario, the worst places in the world with the least funding, the least amount of resources, we can still get something that is a therapy that doesn't produce microvascular complications. I think that's a goal that we can all have. But the one thing, and Peter just noted this very well, the one thing our approach cannot achieve, at least not without implantation of technology, which no one is interested in, that was the first thing Aaron Kowalski asked when he was going, defining all of this, is I can't make it so you don't have to wear something. I have a patient I've been taking care of for 12 years who's getting married next week, and I was seeing her for follow-up. She's been in a couple of my research trials, and I'm finally graduating her now that she's getting married, by the way. And, she said, I really wish I didn't have to wear a pump and a sensor in my wedding dress. And I can't do that. And that is our goal. I agree that it's beyond hemoglobin A1C, it's beyond time and range, and we need to be pushing for those measures. But I just still think it's exciting how quickly we can make gains for those things, where now I can say to the family of a new onset 4-year-old, your kid will never develop microvascular disease. That's not what diabetes looks like in 2026. But I cannot yet say your kid won't have to wear technology. And I think different people will want different forms of therapy. When we were first developing this, I was hooking a kid up on some of the equipment, and the teenager said to me, wow, I feel like I'm turning into a cyborg. And without thinking about it, I said, awesome, because I view that as something kind of cool, but they meant it in a negative context. And so I think we all have our different perspectives on those things.
Well, we are now in our general discussion, and we might just throw in the topic of economics. There was a question related to that. How do we make these products available to everybody, to Greg's patients who don't have resources, all of them, particularly what would be expensive cell-based therapies? Even the closed-loop systems are not going to be inexpensive. But the point is, we need to make products available to everybody that need them, and they need to be affordable, or at least in some way provided by those who care for them.
Silence, Zan.
Well, and maybe that's just where we leave it. We don't have any immediate solutions other than to point out that this is a problem, a challenge.
I'm going to make a couple of comments about that, again, not as answers, but just more as thoughts. So one is, of course, that we are ignoring the huge costs of diabetes. And if you look at this, so the downstream costs over a lifetime, they're huge. And again, if you ask any healthcare system, tell me about dialysis and heart disease, ka-ching, these are huge expenses, but they just seem to be so distal from the intervention that we're thinking about. We often are also thinking about some of these advanced therapies in people who've already got those complications. And, if we have, let's say, for example, we have an expensive cell therapy that's effective and durable, over a lifetime, that could be cost effective. Because again, type 1 diabetes is associated with an average shortage or shortening of life expectancy of 12 years. If you have 12 more years to earn, pay tax, contribute to society, those are huge financial numbers. And so I think, again, generating that data over a lifetime is super, super hard. But we need to have that whole life perspective rather than just, this is a 1-year trial. What did it show in 1 year? And again, the challenge, of course, whenever you have anything new, people will say, well, I don't think it's going to work. Show me the data. So you do the data, you show the trial, then they'll say, I want hard endpoints. So you show them hard endpoints. What did they say next? It needs to be cost effective. So there is this kind of natural resistance to sort of changing the status quo. But again, the status quo is not awesome. Our patients deserve better. And I'm going to keep leaning in.
Well said. And that is the progression. We've got to have an actual therapy before we could begin to do a cost effectiveness analysis. And in the meantime, we need to get them approved and used to get those data.
Can I just make a remark then? I mean, as a type 1 community, we are easily pushed into this whole cost effectiveness discussion. I've been in panels with cancer specialists, and they look at me like I come from a different planet. They never ask this question. I mean, without any scruples, they will defend very expensive therapies if it comes to children with cancer. So I think we need to be a bit more aggressive in our discussions, because type 1 diabetes is not a nice disease. I think on one of Greg's slides, it showed that despite all the kit we give to our people, first of all, not everybody succeeds in achieving time in range above 70% or what have you. Many people still struggle, first of all. Second, it's in the end all about quality of life. And as Peter says, chronic complications, losing your foot, becoming blind, whatever, is horrible. But it's also, even with our new technologies, about the burden of this disease. It's never Christmas. You always have to think. You always have to take your kit with you. You always have to take an extra pump or an extra catheter or an extra this and an extra that. When I talk to people living with type 1, with technology, I always amuse myself by asking, who had an uninterrupted night's sleep? And if there's 50 people in the room, there's always one who says, well, my sensor gave an alarm, my pump blocked, blah, blah, blah. I mean, we're not there yet. And it's about reducing the burden. And there I believe that our disease-modifying therapies and beta cell replacement may be the final answer.
Well said. Over to Jay.
So I'm surprised at all the silence, but well, no, I think that we need to get back and address one other question that somebody raised earlier, and that is what about an Operation Warp Speed? How can we make this go faster, and how can we get it to work? And the only way you can run an Operation Warp Speed is if the government has funds for diabetes research. And unfortunately, what we've had in the last 2 years in the United States is a real cutback in funds for research in general and diabetes research included in that, including the interruption of a gestational diabetes trial, which only had one visit to go to evaluate the final outcomes. And the study was called off, and that visit never took place after much money had been spent to get the trial up to that point in time. We have a government funding situation, which is a disaster. And you can't do an Operation Warp Speed without the government really becoming involved. And the strange thing to me is Operation Warp Speed occurred during the first Trump administration. And Alex Azar, who ran it, he was Secretary of Health at the time. He's now a faculty member at the University of Miami, so I get to see him. And we've talked about it. He has pointed out that they knew what to do to make the vaccines, and they knew how to get the biotechnology going. But the other thing they needed with the vaccines was a distribution system that could get it out to everybody in the country. And they partnered with the United States Army to do that because they had the distribution system to be able to accomplish it. And if they had relied on the trucking industry and so forth, it never would have been able to happen. We wouldn't have gotten the vaccines distributed. So there's always some other side wrinkle that we have to pay attention to. We need government resources to do this. We need investors who are willing to make commitments to companies that are developing these new strategies, and we need to have scientists who are willing to conduct the studies, and we need patients and families who are willing to be research subjects and understand what that involves, and we need regulators who understand what we're doing and can react to it and get these things on the market. Peter has his hand.
I was just going to kind of wonder, and I think Susan made this point in terms of the importance of kind of partnership. And I guess, the question I'm sort of asking maybe the research community in type 1 diabetes is, have we done enough to kind of think how we need to work together? If the stem cell people are excited to be in their little club, and the immunology people are happy to be in their little club, and the technology people are happy in their little club, the risk is that it confuses the outside world. Which one is the answer? Which one is the horse that's going to win the race? You're all competing against each other. When in actual fact, the reality is that, no, we need to play together because we'll each add something which will get us, whatever one across the finish line. So I think that's something that I think about from time to time. I'm not sure that I'm as good. I'm often happy to stay with my friends in my silo, but I think that is something that we need to think a little bit about.
Right. Well, this is precisely what we are attempting to do by bringing all of you who represent these major domains, which are interdependent, as is so clear. So at least I think we make a little dent in that by convening a group of experts who more or less cover the waterfront. And yet the challenges are huge, but the opportunity is out there, and we just need to keep going. I hate to see that we have reached the end of our allotted time, and what a great pleasure it has been to have friends to discuss these important challenges and opportunities, and to have you, the audience, participating. I've seen a very rich chat. I am frustrated that we can't keep that going for the next couple of weeks, but we will at least follow up by providing answers to some of your questions, and we'll hope to get a transcript to you as well as the recording, which you'll get automatically by having registered. I do want to leave on a personal note, and this actually relates to something that Jay mentioned earlier. Many of us were at the American Diabetes Association meeting, and we are all well aware of the controversy. And it's not just in the diabetes community. This was New York Times headline news, or at least New York Times news. It was a very unfortunate happenstance. One of our most esteemed colleagues, Stephen Kahn, was actually evicted from the meeting because of his passing out this paper, which he and other colleagues who we know well had published in Diabetes Care, the organ, the important journal of the American Diabetes Association. Now, the headline or the title of his article says it all. And I recall, Jay, that Stephen was very helpful when we sent in the proposal just a few days after he delivered the Banting lecture 2 years ago. And he was very helpful in getting our commentary published. And so I think this is our opportunity to stand up for science. We're not going to solve type 1 diabetes without it. So with that, I thank you all, and those who can hang around, please do. But this concludes the formal part of our webcast.
Can I make a couple housekeeping comments?
Oh, sorry, Thomas. Yeah, we do
worries.
need to get your parting instructions.
So again, all registrants will receive the link to the recording within a day or so. We want to mention that while we may have 1 or 2 interim events, the Kitalys Institute will hold our first in-person conference since the pandemic, the Asia Pacific Healthy Longevity International Summit, October 1 through 4, 2026 in Hong Kong, with our partner, the APAC Longevity Medicine Society. So please watch your inbox for details. And with that, the formal portion is closing, and thanks to the speakers and you, the audience, for your attendance. Passing the mic back to Zan for our post-panel after party.